The cell is what we typically call life's fundamental unit of structure and function. And what we mean by that is that this is certainly a collection of matter. It will be made up of atoms connected together in specific manners, but it is a fairly self-sufficient unit. There will be a lot of different components of the cell that have individualized functions. There will be parts that are responsible for storage. There will be other locations where we have energy production take place, waste disposal, we'll have transportation systems. So the cell is almost like a little city within itself. Want to direct you to make sure that you watch the little animation that I've posted on Angel. It's an excellent view of the inside of the cell to give you an idea of how complicated the structure actually is. And then what we're talking about in this chapter is typically called cytology, because specifically we're going to be looking at the cell's structure. Now there are two different types of cells. These cells are called prokaryotic cells or eukaryotic cells. If we discuss first the prokaryotic cells, these are what makes up a prokaryote. So that's a type of organism. These are in general what we call very primitive cells. They're very primitive, they're very tiny as well, so they're small in size. And these are from the two different domains, archaea and bacteria. Now you've probably heard of bacteria many times and you're somewhat familiar with what bacteria are. Perhaps you haven't heard of archaea. Archaea are very similar to bacteria, but these are found in a lot of unique environments. So these are found in places where, generally speaking, other organisms are not going to be able to survive. So areas like the deep sea vents where we have a very high sulfur concentration, the hot springs out west where the temperatures are so high. The Dead Sea would be another location, has a very high salt concentration. So we usually don't talk a lot about archaea because they're not in areas where we generally are living at. With the prokaryotic cells, they are so small, so these are not going to be organisms that you're able to see with the naked eye, generally speaking. They're also not going to be what we focus on this semester in this class. We will be comparing and contrasting with them at various points throughout the semester, but our main focus is going to be on the eukaryotic cells. Eukaryotic cells are much more complex. They will also be larger, and these are the organisms that we see around us every day. So these come from the domains first protista. These would be called protist. Protists can't be very large. Some examples of large protists would be the huge algae forests that we see in the ocean, but they can be as small as a single-celled organism. The first picture here is showing us a single-celled protist. That would be a euglena. But we also have amoebas, would be another example of a single-celled protist. In addition to that, fungi would be included in this category. So that would be mushrooms. Animals, humans would be included in that group as well. And then we also have the plant kingdom. I imagine that the majority of you are interested in probably the animal kingdom or the plant kingdom, and those will be our two major areas of focus this semester. Now, first of all, if we talk about some features that you're going to find in all cell types, this would be regardless of whether we're looking at a prokaryotic or a eukaryotic cell. Now, first off, all cells will be bounded by a plasma membrane. That plasma membrane is going to be a phospholipid bilayer, so we should be fairly familiar with what a phospholipid is at this point. And that phospholipid bilayer is just saying that we're going to have two layers of phospholipids connected together. And these are going to be oriented so that all of the fatty acid tails will be on the inside of the membrane. In this picture here, they're shown in yellow. And because that's the fatty acid tails, that will be a hydrophobic region. We could also refer to that region as being nonpolar, because keep in mind that hydrophobic molecules are made up of a lot of nonpolar covalent bonds. The two outsides of that membrane are going to be where we find our hydrophilic head groups, and that means that those sides are going to be very good at interacting with water. Now the plasma membrane, this is the outside covering of the cell, and it is a selective barrier. We want to make sure we use that term selective rather than just say that it's semi-permeable, because it is a choice that is made by the plasma membrane to decide what gets to enter that cell or what gets to leave that cell for that matter. So anything that needs to come inside, whether it be oxygen, nutrients, food, those are going to have to cross that membrane. If anything is going to exit the cell, such as waste materials, they also have to go through the plasma membrane. It is selective because what gets to cross will vary from day to day or from moment to moment, and it is really important that we have some kind of a selection over what gets to come into the cell. Another thing that you find in all cells is going to be cytoplasm. Now, cytoplasm is one term. Cytosol is just another term that refers to essentially the same thing. And what we're looking at here would be an aqueous solution. Recall that aqueous solutions are water-based. And so we're talking about a water-based solution that contains various ions, carbohydrates, and other molecules. So this will be inside of that plasma membrane. All cells are going to need chromosomes or genetic material of some sort. And when we're talking about the chromosomes, we are essentially talking about DNA. DNA is a double helix, so that's not going to change. But the shape of that double helix will vary a little bit depending on the cell type. So all cells are going to need some genetic information to tell them how to make all the various structures that they have, how to respond to different things that may occur in their environment or in their day-to-day lives. And then they're also going to need that material to pass on to their offspring. The last structure that we'll find in all cell types will be ribosomes. And ribosomes, their job will be to make proteins. As we discussed protein structure in earlier chapters, we said that proteins have a lot of different functions inside the cell, and they are the main working molecules. So all cells are going to need some proteins, and they're going to need quite a lot of them. So we will find lots of ribosomes inside every single cell type. Now we want to talk about the prokaryotic cells first and their specific structure. Here we have a typical prokaryotic cell. This would be a bacterial cell, which is certainly an example of one. And this would definitely not be visible to the naked eye. So if we wanted to view a prokaryotic cell, we really would need to use a microscope to do it. And if we're talking about microscopes similar to what we have in the laboratory, we could see the prokaryotic cells in those microscopes, but we wouldn't be able to study too much about them. So other than seeing the overall shape of the cell, we would not really be able to see the interior compartments. Now in the prokaryotic cell, we certainly do have the plasma membrane, as we described earlier. So we've got our phospholipid bilayer that is selecting or choosing what gets to enter or exit the cell. We do have cytoplasm, and that would be all of this tan area here. So that's our water-based solution that's holding all of the molecules. And then the purple that we see in this slide is going to be our bacterial chromosome. That bacterial chromosome, or prokaryotic chromosome in general, is going to be a circular shape. Now this is not to say that it is not a double helix, because it certainly is a double helix, but what we're going to have is we're going to have a big circle of double helix DNA. So as I'm drawing it here, this is in general what it would look like. And then that big double helix is going to be compressed or confined into this area, which we call the nucleoid. So the purple that we see is the chromosome. The area where we see it will be what we call the nucleoid. The nucleoid is not so much a separate structure inside a prokaryotic cell, but it is the location where the DNA is at. The nucleoid is not going to have any outer boundary around it, which means that anything that happens to be in the cytoplasm is going to be able to have access to that chromosome. This is very important to understand, because this is a major difference between prokaryotic cells and eukaryotic cells. We do want to point out that it is a nucleoid. We don't want to get this confused with what we'll later call the nucleus. They are definitely different structures. We do also have a lot of ribosomes in this cell. The ribosomes would be the tiny little brown spots that we see. Notice that they're very small in relation to the other structures that we've pointed out. And notice also that there's a whole bunch of them. So you will definitely see lots of ribosomes inside prokaryotic cells, and then we'll also see a lot inside our eukaryotic cells in just a little bit. There are some other features here to this cell, but in general it is a very simple cell. It's very simple and it's going to be very small. If you go on to take microbiology, you'll talk a lot more about prokaryotic cells because many microbes are going to be prokaryotic cells. Now our focus is going to be primarily on the eukaryotic cells. And if we just throw a slide up here that shows us a typical eukaryotic cell and a typical prokaryotic cell, the first thing that we should notice is that there is a major difference in size between the two. If we look at the prokaryotic cells in our microscopes, we can't see a whole lot more than the overall shape of the cell. But if we look at a eukaryotic cell in our microscopes, not only can we see the shape of the cell, but we can distinguish a lot of interior compartments, and we may be able to see some details about those interior compartments. So now we want to focus on the eukaryotic cells, talk about what makes them different from the prokaryotic cells, and then talk about the individual components that they do have. So first off, the eukaryotic cells are going to have extensive and elaborately arranged internal membranes. These internal membranes are definitely going to be phospholipid bilayers. We'll see that all membranes are phospholipid bilayers, and we'll talk about that in detail in Chapter 7, but they're going to have a lot of inside membranes. So they will have the plasma membrane, which will be around the outside of the cell. So this will represent my plasma membrane. But then they additionally have a lot of membranes inside. What these inside membranes are going to do is they will divide the inside of the cell up into structures which we typically call organelles. You can think of these similar to the organs of an animal. So each of these organelles is going to have a specific function. So now we have little compartments that can specialize. One of these compartments will be called a nucleus. So the nucleus is just one example of a specific organelle. We'll talk about its specific function, but the term eukaryote means after the nucleus. So that nucleus is certainly an important part of a eukaryotic cell, and we're going to see that with few exceptions in every single eukaryotic cell. So we want to make sure that we understand that there are lots of inside membranes with eukaryotic cells, and that they're going to give us a lot of compartments that can now specialize. Typical examples of eukaryotic cells are animal cells and plant cells. Those are certainly the two that we're going to be focusing on. If you look at this slide, it shows us an animal cell. Hopefully we can see that there is a lot more stuff inside this cell compared to what we have with the prokaryotic cell. The nucleus is the big purple structure right there in the middle. It is a membrane-bound compartment, so it has a membrane on the outside, but there's also a lot of other membrane-bound compartments. We will talk about those various compartments as we go through this chapter. We'll talk about their overall makeup, and then we'll talk about their specific function. The plant cell is going to be very similar to the animal cell. We will have some unique structures in the plant and the animal cells, and then we'll have a lot of common structures between the two. They are both eukaryotic cells though, so this means that they will have a lot of internal membranes or membrane-bound compartments, which we can call organelles, inside of them. So we're going to start going through and talking about those individual components, and since eukaryotic cells are really named for the presence of the nucleus, we want to start off with the nucleus. The nucleus is, generally speaking, the most conspicuous organelle inside a eukaryotic cell. And what we mean by that is this is usually the largest organelle, and it's the easiest one to see. A lot of times, if you look at a eukaryotic cell in a microscope, you can see the nucleus without even really having to stain the cell if you properly adjust the light. So it is fairly easy to see, and this is going to be where most of the genes are found in a eukaryotic cell. So by most of the genes, we're saying most of the DNA is going to be located inside the nucleus. This nucleus is going to be enclosed by a nuclear envelope, which is the membrane. And in this case, it actually happens to be a double membrane. So 2 phospholipid bilayers that are going to be surrounding the outside of this nucleus. This will then isolate all of the insides of the nucleus from the rest of the things in the cytoplasm. So here we have a bigger look at the nucleus. That nuclear envelope is what we just introduced, and that is 2 layers of membranes. So 2 phospholipid bilayers. It's called the inner membrane, would be the inside one, and the outer membrane would be the outside one, obviously. And then inside is where we're going to find all of that DNA. Keep in mind that the DNA does hold the genetic information for the cell. Not only is this material that is going to be passed on to future generations, but it's also information that is necessary to run the cell, and that's run the cell on an everyday basis. So we certainly need to be able to access any information that happens to be inside the nucleus. So we want to point out that on the surface of the nucleus, on that nuclear envelope, there are what we call pore complexes. These pore complexes are going to be little protein structures that provide essentially a little tunnel that molecules can both enter the nucleus and they can exit the nucleus. Now, this is not to say that it is an unregulated entry and exit. It will certainly be regulated. This means that not just anything can pass back and forth through these little tunnels. These pores will be able to open and close to select what gets to cross that membrane. So at various times throughout the day, throughout the seconds, throughout the moments, we're going to have molecules that both enter the nucleus to look at the genetic information there, and then we'll also have molecules that exit the nucleus to take that information to the rest of the cell. Another important structure of the nucleus is the nuclear lamina. And the nuclear lamina, you could list a lot of functions for it, but one of the most important functions that the nuclear lamina is going to have is going to be organization. If the nucleus contains the vast majority of the DNA inside the cell, it has a lot of information inside it. If we just focus on human cells are estimated to have 25,000 genes inside their nucleus. Now that's based on information from the Human Genome Project. That's a lot of genes. On an everyday basis, a cell is not going to be using all 25,000 of those. So one cell may be using 3,000 of those 25,000 genes. Well, if you needed to find gene number 150, we would want to know where that's going to be located at, because we can't spend time sifting through 25,000 to find just one. So the nuclear lamina is going to be one way that we really organize these genes, so that when we go in there to find a specific one, we can find it very quickly because we know exactly where it's going to be located at. So the nuclear lamina is a structure that is found just inside the nuclear envelope, so it would be right around here, and it's also found all throughout the inside of the nucleus. So it's like a net that runs throughout the inside, and this is going to provide a lot of attachment points. So this will allow us to attach specific genes to certain locations within the nucleus. Again, so that when we go in there, we know exactly where we're going to find gene number 150, gene number 500, and so on. Another thing that the nuclear lamina is going to do for the nucleus is that it's going to help maintain the nucleus's shape. So generally speaking, the nucleus is going to be fairly spherical. So this is going to help us make sure that it does stay pretty spherical. We will also have what's called chromatin inside the nucleus. The chromatin is what we refer to the chromosomes as being made of. Chromatin is DNA and protein associated together, and that's the chromosomes. Chromatin is kind of a generic term that refers to the DNA and the protein, And then if we talk about a whole piece of chromatin, then we use the term chromosome. So we do want to kind of see the difference between the two of those. When we're talking about chromosomes inside of a eukaryotic organism, these are going to be linear chromosomes. So now instead of that big circle of DNA, we're going to have these linear pieces like this. So we want to make sure that we understand that we do have linear chromosomes. In a typical human cell, you're going to have 46 chromosomes. So these 46 chromosomes will all be inside the nucleus. Additionally, there is a structure called a nucleolus that stains very darkly inside the nucleus. There may be a few of these inside of a nucleus. So by few, maybe 2 or 3 of these are visible inside the nucleus. But it's important to understand that this does not have a separate boundary around the outside of it. It stains a little darker just because this is a location where you do find a lot of ribosomal RNA. That means this is the location where the ribosomes are going to be put together or manufactured. And because they are manufactured there, or they're in a high concentration in that one location, it tends to stain fairly darkly. As far as the overall functions of the nucleus, some of the functions should seem quite obvious at this point. It is going to store the genetic information of the cell. So this is like a little filing cabinet where all of the information will be. It will be highly organized so that we can find whatever information we need to find, and we can do it quite quickly. Additionally, because all the information is there, it is also going to be responsible for controlling the activities of the cell. So in that way, you can think of it as the brain of the cell, or the CPU, if you want to think of a computer of the cell. The way that it's going to do this is it's going to synthesize some messenger RNA or mRNA, and it's going to then control or direct the protein synthesis. That specifically is what we'll look at in chapter 17. So we're not going to go into that particular function right now and talk about the details of it, but we do want to know that the nucleus is responsible for controlling the activities of the cell. With few exceptions, you will find a nucleus in every single eukaryotic cell. And again, that is really where that term eukaryote comes from. Make sure that you can see the difference between the nucleus and what we previously described as the nucleoid. The nucleoid does have chromosomes in it, but there is no outer boundary. Because there is no outer boundary or nuclear envelope around it, there will also not be pores, there will not be a nuclear lamina as well, and you won't have that nucleolus region either. The next part of the cell that we want to talk about are the ribosomes. And the ribosomes were one of those things that was present in all cells. So this would mean both eukaryotic and prokaryotic cells. These are going to be made up of 2 different types of molecules. They will be made up of some RNA, which, because it's found in the ribosome, we call it ribosomal RNA or rRNA. And then they also have a protein component. The rRNA and the protein are going to be wrapped around each other and assembled together into some subunits. We will have what we call a large subunit because it's large, and we'll have a small subunit because it's small. And those 2 subunits will come together when the ribosome is actually functioning. If the ribosome is not working, these are going to be actually in pieces inside the cytoplasm. So we do have 2 different locations where we do find the ribosomes. They can be what we call free ribosomes, and those are basically floating in the cytoplasm. And then we also have some ribosomes which are going to be bound ribosomes, and these are bound to membranes. Those membranes may be the nuclear envelope. We'll also have some other membranes where they'll be bound. The ribosomes are extremely important to a eukaryotic cell because these are the ones that are going to be performing protein synthesis. By performing protein synthesis, they are constructing that primary structure of the protein. So recall when we talked about the primary structure, we have to get all the amino acids hooked together in the correct order. If we are hooking together the amino acids in the correct order, essentially what we are doing here is we are making peptide bonds. So the ribosome will be making the peptide bonds of the primary structure. Ribosomes are quite small, and you will have literally hundreds of these, if not thousands of these, inside a single cell.